Okay, hello everyone. I do have 2 p.m. Central Time here, so we will go ahead and get started. Thank you everyone for joining us for the Soil Health Nexus Digital Cafe series. For those of you who may not be familiar with the Soil Health Nexus, we are a 12-state university-led team dedicated to increasing access to research-based soil health knowledge, uh, research, and extension. We are an extension-based team, and we have representatives from all 12 North Central region states. And we're really dedicated to increasing the resources and education around soil health for extension educators. So, as a part of that, we host this digital cafe series monthly where we feature experts around the region, and I'm excited to feature Ganga Hettiarachchi today. We also host trainings, and I do want to let everyone know that coming up next month, October 23rd, we are collaborating with Iowa State University to host a training on soil health and livestock and the below ground benefits of soil health, or excuse me, the below ground benefits of livestock on the land. That's going to be in Ames, October 23rd. And it is, while it's based in Ames and hosted collaboratively with Iowa State University, it is a multi-state event, and we do encourage educators and soil health professionals from across the region to attend. I will put a link there for the registration and for more information on that event in the chat. Okay, so we will dive in here and get started. What we're going to do today is have our presentation, and then we'll have Q&A. And then if time allows, this is a 1 hour session, but if time allows, we will have breakout rooms. And those breakout rooms, we have them in those five categories here, so folks can kind of join the breakout room that makes the most sense to them and talk with their colleagues about the presentation, about the research shared, and how this might influence your work or how you might use this new knowledge moving forward. Today's talk is on the impact of organic amendments on carbon stabilization and soil aggregates, and I'm very excited to have Dr. Ganga Hettiarachchi here from Kansas State University and the Department of Agronomy there. Dr. Hettiarachchi is one of the leading scientists in the fields of trace metal and nutrient chemistry in soils. So very excited to have her here to talk to us, as well as talk to us about a topic we hear a lot about, soil aggregates or organic amendments, and what is the impact of that? So I think this is an outstanding question that a lot of folks are having as organic amendments become more popular. And so excited to hear a little bit more about this research. I will stop sharing and hand it over to you, Kinga.
Thank you, Anne, and thank you everyone for giving me opportunity. Let me share my slides. Can you see my slides?
Right now, we're seeing presenter view, so I think you might need to do that swap. Presenting
Okay,
slideshow.
let me see what I can do. Stop sharing, and then is it still the same?
Yep, looks great.
Looks great. Um, So good afternoon, everyone. So I'm going to share some of the results from a study conducted at several different places. I will explain it to you in a bit. And then for this talk, I'm going to focus mostly on the impact of organic amendments on carbon stabilization in soil aggregates. Little bit about the background. I don't think I do need to tell this group, but you will know that the soil organic carbon, that's a foundational component in soil. We have numerous benefit from soil organic carbon or soil organic matter. And then soil contain the largest inventory of organic carbon on the Earth's surface. And then if we look at globally, and then the soil carbon and what's in atmosphere and biomass combined, and then we know that the soil contain more carbon than those two reservoirs combined. So with that, soil carbon stocks are a significant carbon sink. And it's not just like taking carbon out of atmosphere, but you do know that when we do have in soil, that's actually going to benefit us in various ways. So a little bit about if you're thinking about increasing carbon dioxide levels and then looking at this same thing as an additional thing, like how we can, while you are increasing carbon stocks, how you can improve the environment as well, or help the environment as well. We can look at the sixth IPCC sixth assessment report from Working Group 3, 2022. And then it talks about the various sectors and talking about mitigation options in sectors and systems. So agriculture is one of the systems, and then it is the one that actually second largest sector that capable of doing significant carbon dioxide mitigation. So the other sectors are the energy, infrastructure, transportation, forestry, etc. So the soil organic carbon. So if you're thinking about this as an option, so our agricultural practices collectively can make a significant contribution to the global climate change mitigation. And how we can do it, improved and sustainable crop and livestock management practices. We are going to talk about it, one of that today. And then the increasing carbon sequestration and then using renewable energy, etc. So we know that soil can act as a sink, but at the same time, if not properly managed, soil can be a source as well. So soil can remove, if we think about the estimated, like annually, carbon from the atmosphere, about 0.4 to 8.6 gigatons of carbon. And this can counterbalance about 10 to 15 of global fossil fuel emissions, just to think about the significance of this. So how can we manage soils and how can we actually increase soil carbon storage? What are the management practices and the conservation or the no-till practices and adding organic amendments? Let me get this, hide the floating panels so that I can see the slide. So adding organic amendments and then adding things like, including things like biochar, the crop rotation, and then also the intercropping as well, because when you have the different types of crops at the same time, that could be beneficial as well to carbon sequestration, and then the cover crops. So you all are familiar with these uh, uh, practices. So if we look into like how we can conserve soil organic carbon and how soil organic carbon is preserved in soil, we can look at like three different ways of doing that. So earlier we were totally focusing on the what we call as biochemical recalcitrant. In other words, if you do have recalcitrant organic carbon, then those will not be subjecting or easily degrading. So that would be a way to preserve it. But now we talk about that if you provide right conditions to soil microorganisms, although slow, they could, even these recalcitrant carbon, they could decompose. So now we more and more understand the other types of carbon preservation. One is physical protection. So that is, if you're thinking about like they're having carbon where that in a place that microbes cannot easily access. So physical protection, like minerals can physically, soil minerals can physically protect organic carbon from degrading, decomposition. And then the last, but the another important mechanism is chemical stabilization. We know that the organic carbon, even the soil organic carbon, can be adsorbed or sorbed onto soil particles, especially the iron minerals, calcium bridging to other minerals through calcium bridging, etc. So chemical stabilization can happen so that they can be preserved in soil for a long time. So we know that some of these carbons can be like protected for long time in soil. So this study is about looking at the soil carbon stabilization mechanisms in situ in free soil microaggregates collected from different managed temperate ecosystem. So in this study, we actually had two different sites where we focus on soils from a temperate continuous cone system established in 1990. We sampled that in 2012, so after 22 years after establishment. And then we also, I'm not going to talk about that much, we also focus on a tropical agroecosystem where tillage and the crop rotation was the management practices. They had simple crop rotation and then also complex crop rotation, and then looking at how simple versus complex crop rotation can impact carbon sequestration. In this the study, we were more interested in looking at the trying to understand how they are protected in aggregates and like visually without disturbing them. So the Kansas one is the one that I'm going to focus on. So the samples were collected for this study 22 years after establishment. So it was a molly soil located in Manhattan. So we know that the soil organic matter is the something we still don't understand, and it is very complex. So because of this, we actually integrate various approaches to do this study. So we use traditional wet chemical methods, and then we also use different extractions, like separating humic acid, and then characterizing those using 13 CNMR, HPLC. And then we also, like we traditionally do, look at what are the total organic carbon level differences, what are the labile carbon, reactive minerals, etc. And then we use two other techniques that allow us to look at carbon in different way. One is using bulk X-ray, near-edge X-ray absorption, fine structure spectroscopy, that to get like an idea about bulk speciation of organic carbon. If you do have a sample, representative sample, what's the speciation look like? And then I am not going to focus on that part today. And then I am going to focus on the study we did with what we call as a spectromicroscopy, X-ray spectromicroscopy. It's a long acronym, STEXM Nexus. So it is standing for Scanning Transmission X-ray Microscopy, combined with near-edge X-ray absorption fine structure spectroscopy. Let me put this in simple terms. What this means is we can, if we do have the right energy range, we can look at carbon or like other element of interest, like we can map them while we are getting their chemical information. So basically, that's what we did. And then this will give us a like a spatially resolved type of a carbon maps and then other elemental maps as well. And here, this technique can also, you could do this like submicron level. So you can see that why we needed to combine this with wet chemical analysis and the other bulk techniques, because we have only limited capacity to collect information using the spectral microscopy. You cannot use like, you know, 20 different aggregates or 100 different aggregates from one system. So that's the disadvantage of this approach. So a little bit about soil sampling and sample preparation. Soil samples from a long-term, as I said, established in 1990. It was a continuous cone system. So you could consider this as a probably like a pretty bad system when it comes to carbon sequestration. You have a monocropping for a long time, and then nothing else is there. So we had a split plot randomized complete block design with 4 blocks, and main plot represented tillage, conventional till and no-till. I'm going to focus on no-till here. And then within those no-till, we had these subplots uh, receive different nitrogen treatments. One is no external input, that's the control. And then the second is organic nitrogen fertilizer. That one, first 10 years, it was as a cattle manure. And then the subsequent 12 years, it was composted manure, the food waste, as well as crop residue. So that's what the organic fertilizer treatment. And then the inorganic, that was urea. And then the same levels of nitrogen was applied, 168 kilograms of nitrogen per hectare. And then for organic nitrogen fertilizer, we considered that all inorganic nitrogen, 100% was available. And then the organic nitrogen, 35% is going to be available per season. So the data collection, the sample preparation is tedious, and then it's more tedious because we wanted to preserve the aggregate structure, architecture of the aggregate. So what we did was we chose by sieving, wet sieving, we chose 150 to dry sieving, sorry, 150 to 250 micrometer microaggregates, and then saturated those for 16 hours using a humidifier with aid of a humidifier. And then snap freeze this in liquid nitrogen. And then we use a cryo ultramicrotome to cut this at 55 degrees Celsius so that we could get like a thin sections preserving the architecture of the aggregate. And then so this is the PhD student work on this research. Pavitra is working on that. And then once we have this section, carefully that into a TEM grid, and then store it in a helium environment. And then we do need to take this to synchrotron facility to conduct this work. We went to Canadian Light Source, or the part of this work was done at the ALS, Advanced Light Source, over in Berkeley, California. So data analysis also tedious, but I'm not going to go into details. And then just to show you results here. So this is showing what we call as cluster indices map of carbon. So just to give you some idea about, so this is from the no-till control, no-till organic fertilizer and no-till inorganic. So if you're looking at this first map with multiple colors, so those are really, cluster map is basically giving us the carbon distribution in the area we looked at. And then different colors shows that the areas with similar chemical characteristics. So for example, when we are looking at no-till control, we could, through cluster analysis, we could identify 4 different areas sharing similar chemical characteristics. So this is, when it comes to carbon, this is most likely combination of many different things together, but sharing similar chemical characteristics. And then you can see the other maps as well, the organic and as well as the inorganic fertilizer added one. So moving on to the so when we are looking at, we can actually, for this each of these cluster map, we can actually obtain what we call as a Zane spectra. The especially like that spectra tells us about the chemistry, what kind of carbon we have. We have different features that we can identify, for example, presence of peptide bonds and things like that, telling us it is a microbially derived carbon, or looking at certain species and then telling us it's a plant derived carbon. So likewise, we can get rough idea about what kind of carbon we are looking at. And then the again, so just to give you that how we can understand the different types of carbon we have here. And then here, this one is showing the calcium map. Again, similar interpretation. So this is identifying about 3 different areas in the NT control, and then the organic fertilizer added one, or the manual compost identify 4 different clusters, and then the inorganic fertilizer added one identify 2 different clusters. Again, the once we have this, we can extract the spectra, and then X-ray absorption spectra will help us to identify the chemical speciation. Most of the time, even if you are looking at like a very microscopic, almost submicroscopic pieces, still you would see that rather than having just one calcium species, it is still a combination of species. So, and then this is the showing the iron map. Iron map was only collected for the NT control and the NT no-till control and the organic fertilizer. Again, showing that you could extract a spectra and look at the speciation. So the what when you are looking at map, you can also look at like how these are distributed. Are these distributed like evenly, or are those concentrated in certain way? And then sometimes we can recognize that the carbon might be in like a small, the nanotube kind of like a part within minerals, like preserved in there. So we can get a lot of information if we are able to collect several different maps as that. I will show you some example actually clearly showing that distinct coatings of iron around the microaggregate. So again, I am not going into details, but we can look at the iron, what kind of iron chemistry we have. So once we have this map, we can also look at something called as chemical contrast map. Basically, we can separate different types of carbon. We can separate calcium, we can separate iron, etc. So you can see that when you are looking at this, the same piece and looking at carboxylic carbon and calcium and then iron, so especially calcium and carbon, you can see the intimate kind of association between those two. You do have similar kind of association with iron as well. But at the same time with iron, mostly you see that it's like mostly like coatings or like protected the calcium, carbon compounds within those. So that was another thing that we were visually able to see from undisturbed cross section of a microaggregate. So again, like I said, those are really nice, really like extremely useful information we can get. But at the same time, to get a better idea, we need to integrate this with bulk chemical analysis. So we did take those samples. We had four field replicates. So take those samples, and we did like looking at the total organic carbon, and then also looking at the extractable ammonium oxalate extractable iron, aluminum, and silicon. So we can see that the clearly we had more than double in some cases compared to control, almost like a triple levels of organic carbon in this organic amended soil. Going back, and then at the same time, when we are looking at the ammonium oxalate extractable, ammonium oxalate is this specifically designed to extract the amorphous ion, and then it extract aluminum, silicon as well, other things as well. So looking at ammonium oxalate extractable ion, you can see that clear, the significant enhancement in ammonium oxalate ion, significant enhancement in ammonium oxalate extractable silicon. And then the aluminum also higher than the control, but not higher compared to the fertilizer added one. Again, indicating that the higher concentration of SOC and this higher concentration of almost like amorphous minerals we can see. And I'm my apologies about the sound if you are hearing. That is something that we have in the building. So sorry about that. And then looking a little bit to get a better idea, looking at ammonium oxide extractable fraction of iron, aluminum, silicon with the total organic carbon percentage. So you can see that the clear like a relationship between these two, the extractable fractions and TOC. So suggesting that the enhancement of these fractions actually enhancing the carbon storage or the carbon retention as well. It is not just our studies, other studies have found too, including soils like paddy soils, that organically amended soils contain significantly higher concentration of poorly crystalline iron. And then we know that these poorly crystalline ions are iron minerals, are very active, reactive, so they are contributing to carbon storage. So the soil organic carbon storage is closely related to that. So as a summary, So the no-till manual compost addition enhances both carbon sequestration and stabilization. And then we do think that those mechanisms are a combination biological, chemical, and physical stabilization as well as protection. And then I have not shared that, but we have seen that the protection of carbon compound, preserving even the morphology of plant or microbial structure within microaggregates. One of the maps we did see like a root hair preserved as is, and then when we look at the chemistry, we can see that it's resembling, closely resembling plant-derived carbon. So the intimate association that you observe, like preserve carbon and soil minerals, such as chemical protection. So sometimes at the same time, mineral coatings most likely make a physical disconnection between plant material, the organic carbon and microorganisms as well, so making it protected that longer for that way. And then calcium contribution is also clear from our work that, and then that's a mechanism suggested by many that how the calcium could really have like a bridge between organic carbon and the soil minerals. So with that, I want to thank Pavitra. So most of the work that I presented, actually Pavitra's PhD work. And then my collaborators and contributors, including Dr. Rice, as well as Chammi, and the different collaborators from K-State, and then some are from either from the Advanced Light Source or the Canadian Light Source. And we had REU students, undergraduate students working on this project and helping to develop certain methods and did some data collection with wet chemical methods, Dorothy, Allison, and Noji. So thank you.
All right, great. Thank you so much, Ganga. We really appreciate you taking the time to share some of your work. For everyone on the call, what are your questions? As they've come in, you can either put them in the chat or you are welcome to unmute and ask them yourself. All right, well, if there are no questions, quiet bunch here today, we can go ahead and go into the breakout rooms to allow for some discussion. So I have opened up those rooms and folks can choose the breakout room that makes the most sense for them. So we have educator, agency, student, farmer or rancher, and then NGO. So feel free to choose the room that makes the most sense for you and join that room. You can also join a room that maybe you're not in. I've certainly sat in on the farmer rancher group before, even though I don't own land myself. So you also can take that opportunity as well. Ganga, are you able to share to stick around? Usually we have a researcher room as well. I apologize, we don't
Yeah,
at the moment.
I can. There's a question like any surprises with the results
Oh yeah,
in the
from
chat.
Valerie. Great. Thank you for seeing that.
Yeah, yeah. So actually no, but we were kind of surprised that seeing a root hair that you would think that it would not be something like a preserve in an aggregate for a long time, but seeing some resembling those kind of things were Not unexpected, but at the same time, it was kind of like uh, I mean, felt really good of like seeing it there.
Great, thank you. Any other questions coming through? All right, well, those breakout rooms are open. You should be able to go to the bottom of your screen here and look at the breakout rooms and then choose the one that makes the most sense for you. Kinga, thank you for sticking around. I might put you in the agency room if that's okay, because we do see we have folks. Uh,
Sounds good.
okay.
Please, I'm fine with any group.
Thank you. I apologize. We normally have a researcher group, but I don't see that one there. All right, I'm just going to give a few minutes here for folks to join.